What is molecule?
Molecules means like proteins.
Of course, they are the example. Protein molecule, right? Carbohydrate molecule, right? But how can you define?
The element essential for the life we call molecule, sir.
Element essential
For life, living beings.
Okay.
I think so, sir. I don't exactly. I don't.
Okay, somewhat right.
Group of atoms, like chemistry, it is
Exactly.
like
Exactly. Group of atoms. Group of atoms bounded together to form a molecules or compound, or it is called as chemical compound, right? Then what is atom? You said the group of atoms, right? Molecule means group of atom. Before that, one of your student, your friend said the molecule example protein, right? And one of your friends said the molecule means group of atom. My next question is, what is atom?
it is the smallest unit which cannot be divided further.
Right, atom is the right smallest unit. Okay. And another definition, is there anyone? Simple, it is the basic unit, basic particle of a matter.
Basic
That is
building
most basic level which contain at least one proton. In an atom, protons, electrons, neutrons also present. Even it is only one proton present, that is also considered as the atom. Right? So can you give any example for atom?
Hydrogen atom, oxygen atom.
Right, hydrogen atom, oxygen O2, hydrogen carbon atom, neon Ne atom, argon air atom. So they are the atom. The molecule means group of atom. Give me example. Molecules.
Proteins, nucleic acids.
Right, of course, protein, nucleic acid. How they are made up of? What are the main elements?
Amino
or
acid.
main atom?
Amino
acids. Amino acid formula.
Sugars and
Of course, sugars also. Okay, right. You said amino acids. Can you tell amino acid formula?
COH, some RX.
Amino acid NH2, nitrogen, hydrogen together form amino acid.
Yes, sir.
So nitrogen atom, hydrogen atom, these two atoms combinedly form a molecule NH2. That is a simple molecule. And the group of molecules, that means group of NH2, altogether form a compound molecules. That's what we studied in molecular biology. So especially biology, molecular, of course, I hope you understand about the atoms and molecules, right?
Yes, sir.
Then what is biology?
Which deals with the study of plants and animals.
Plants and animals, not only plants and animals, any living,
living organisms.
any living creature,
Basically,
any living organism. Then how can you define whether it is living or non-living?
By the presence of the cell molecules.
Cell line nucleus.
Right, cell molecule, especially the okay, right, presence of cell molecule, right? What are the cell components?
Nucleus, mitochondria, Golgi apparatus, lysosomes.
Endoplasmic reticulum.
In order. Can you tell in order?
Cell wall. Cell
Plasma membrane,
plasma membrane,
mitochondria,
Okay.
Golgi apparatus,
Okay.
Endoplasmic reticulum, smooth reticulum,
endoplasmic
nucleus.
reticulum,
Right.
lysosomes,
Okay. Right. Are you visible to screen? Screen
Yes, sir.
visible to you?
yes sir, yes sir,
Yes, sir.
Right. So cell, first we started from the molecule, means group of atom, and also we know the atom, what is atom and examples of atom. And the atom formed together formed a simple compound and compound, so that means simple complex and compound complex, so on. Let's see how they are made up of, how we involved in biology. So in biology means, you said any living organism, study of living organisms such as virus, bacteria, plants, animals, so on. So the living nature, the based on the cell components, not only one cell components, most of the characters to living, the most of the one, first one is movement, and it should be reproductive, and it should be cellular components, and especially genetic material. If any cell with genetic material, that is considered as a living. Without genetic material, non-living, because all character present inside the genome, right? Before that, we should know about the cell components. In plant cell, clearly two components. One is cell wall, right? Cell wall. The rest of the all membrane, rest of the all part called as protoplast. Protoplast. We know cell wall made up of cell wall. So no need here. No need to explain here. And what are present inside the protoplast? What are present inside the protoplast? For example, in order, this is, for example, this is the cell. Cell wall completed. Cell wall. Now the rest of the place. Now the entire portion. This entire rest of the portion called as protoplast. Right? And again, protoplast classified into protoplasm and cell membrane or plasma membrane. Plasma membrane. Plasma membrane or cell membrane. And next one, protoplasm. Don't get confused. Protoplast. Here protoplasm, right? So only this layer, plasma membrane. Only this layer, only the second layer plasma membrane. Except the plasma membrane, the rest of the all called as portion. This
Protoplasm.
right, protoplasm, right. Again, protoplasm classified into nucleus,
site,
right? Nucleus and cytoplasm.
cytoplasm,
Nucleus and cytoplasm. Here we can take this blue color, nucleus, right? Nucleus completed. Now what is the rest of the portion? That means around the nucleus occupied by, that's it, cytoplasm. What are present inside the cytoplasm?
organelles,
Cell
ribosomes.
organelles. All cell organelles present, and further divided into cytosol also. Cytosol just liquid without any cell organelle, only just liquid and minerals called as cytosol. They are the clear in order cell components. The each and every component, cell wall made up of molecules, plasma membrane made up of molecule, protoplast molecule, protoplasm molecule, nucleus also molecule, right? But already told you the clear living nature based on genome, right? Based on genome. Now, where is genome? Where is genome?
Chromosomes in
right chromosome. Then the location of chromosome,
inside the nucleus.
Gene.
right inside the nucleus.
Yes, sir.
Right. So nucleus is the part of cell, right? It is the center, occupies the center position. That is the nucleus, right? Is it clear? Hello?
Yes, sir.
Yes, sir.
Right.
Yes, sir.
You said inside the nucleus chromosome present. Can I rough this?
Yes, sir.
Can I
Yes, sir.
rough?
Yes,
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 sir.
sir.
Okay. Thank you. Nucleus. Inside nucleus chromosome, right? Chromosome made up of chromatin. I will tell you later what is the chromatin. And inside the chromatin, DNA. Inside the chromatin, DNA. Right? DNA further classified gene. This is the clear concept. Can you differ DNA gene? What is the difference between DNA and gene?
Gene.
క్రోమోజోమ్స్ పైన డిఎన్ఏ ఉంటుంది, డిఎన్ఏ పైన జీన్స్ ఉంటాయండి.
Of course, right, but clear definition. That's also right.
Heredity materials. Gene.
Gene is heredity material. Okay. I am asking how can difference?
Gene is a
DNA,
part of
gene.
DNA.
That's it. Gene is a part of DNA. That means
DNA is called gene.
that DNA is the elongated strand. It included active and non-active regions. Active, non-active, both are present inside the DNA. But only active region, only active portion called as gene. Active portion of DNA, only active portion of DNA is called gene, right? So the group of genes called genome. So our first class is genome organization. How the genomes are organized in higher plants or any animals and any organism. Of course, in examination may be given genome organization, simply genome organization or genome organization in plant. Whatever it may be, the almost all same, the same type of makeup, genetic material in all organisms. That means here, for example, if you take the genome, can I rough this?
Yes, sir.
Hello?
Yes.
Yes, sir. Yes, sir.
Yes, sir.
Right. Our first unit genome organization, right? First unit. What is the first unit?
Genome organization.
Right. Genome organization. Genome organization means how the genes are compacted inside the cell, how the genes are arranged inside the cell. Let's already told you cell, nucleus, chromosome, inside the chromosome chromatin, inside the chromatin DNA, and finally genes.
DNA.
Finally genes are present, right? Let's see the genome organization or chromosome organization. Let's see first. Just wait. Before that, we should know about the chemical composition of chromatin, because nucleus, chromosome, already told you chromatin. So genomes or genes packed inside the chromatin. and this group of chromatin together formed as a chromosome, which is packed inside the nucleus. So chromatin here clearly said the chromatin made up of chemically it is made up of mainly 3 different chemical compounds. One is DNA, and second one is proteins. Third one is RNA. DNA, protein, RNA. Right? So we want DNA only. Right. Then again the protein, 2 types of protein present in chromatin. One is histone protein. Second one non-histone protein. Non-histone. And RNA. These substances are present in chromatin in different ratio. These substances. Of course, we know genome organization. Genome organization means what are the present along with the genome? What are the present along with the DNA? Here the DNA, you know very well the genome DNA packed in chromatin. How the chromatin made up of? The simply chromatin made up of 3 compounds, such as 3 chemical compounds such as DNA, protein, RNA, right? But they are arranged in different ratio. Generally, histone proteins and DNA 1:1 ratio. Histone protein, this histone protein, just wait. This histone protein and this DNA arranged inside the chromatin 1:1 ratio, right? While DNA and non-histone protein and DNA and RNA equally arranged, that means 1:0.6. DNA 1 common, already told you. Non-histone protein less than histone protein, that is 0.6. Here RNA also 0.6. That means in chromatin arrangement DNA 1%, protein histone protein 1%, and another chemical compound non-histone 0.6 and RNA 0.6. Of course, it's somewhat less when compared with non-histone protein also. In this chromatin, mainly highest DNA histone protein and very less amount RNA. Right? This is about composition, chemical composition of chromatin. Let's see about in detail about the proteins. Proteins already told you, histone protein and non-histone protein. Right? First of all, in this chromatin, almost 60%. Wait, just wait. 60% protein. Rest of the portion DNA and RNA. Right? The proteins play very significant role in organization. That means in genome organization. What is organization first? What is organization? What is genome? Already told you genome and gene definition. Gene is the active site of DNA, right? Then what is organization? Hello?
Systematic solution.
Right. Systematically arrangement. Where it is systematically arranged? It is systematically arranged in a chromatin threads. The chromatin threads together formed chromosome structure. The chromosome packed inside the nucleus. The nucleus part of the cell. Right? Let's see how they are packed. Actually, this genome packed with help of proteins. Let's see how they are packed. Can I rough it? Hello?
Yes, sir.
Yes, sir.
let me complete one by one. First of all, major portion proteins, that mean histone and non-histone protein. Almost 60% chromosomal constituents, chromatin constituents. The proteins play a very significant role in organization of DNA and also replication of chromosome. I will tell you later. The proteins present in the chromosome are chromatin threads, two types already told you, namely histone and non-histone. First of all, let me complete histone protein. Histone proteins. Histone proteins. Histone protein. These proteins are also called short as histones. Simple shortcut histones. These are alkaline nature. They are firmly, tightly attached with acidic nature of dash. What is acidic nature? Acidic nature of DNA. Acidic nature of DNA. So histone proteins are alkaline and DNA acidic nature. The histone proteins in eukaryotes are 5 different types. The histone proteins 5 different types such as H1. If you possible, write down one by one. H1, H2A, H2B, H3, H4. So these 5 types: H1, H2A, H2B, H3, H4. The most interesting event is the histone proteins are highly conserved components. They are highly conserved from generation to generation. That means in a pedigree, our forefathers also same histone proteins, and before forefather also same histone proteins. There are no change in histone proteins nature. Long evolutionary history, they are constant. These proteins play an important role in maintaining sequence of amino acids in a protein without much alteration of generation. Without much alteration. So the histone is protein, right? How the protein made up of?
Amino acids.
Amino acids.
A group of amino acids. Group of amino acids. I will group of amino acids. So the group of amino acid order, for example, in a cell, 21 type of amino acids. They are arranged in different manner. For example, A, that means this is A is one person and A also another person. This is the ancestor. A is the ancestor, small a. Capital A is the offspring. The ancestor histone protein sequence amino acids. For example, first A amino acid, type of A, B, D, Z, X, C, like this arrangement. The same sequence of amino acid arrangement, A also same continue. A, B, D, Z, X, C. So that means it's much conserved. Histone proteins are much conserved, no change in evolutionary. Right? They maintain the constant. This conservative approach of histone proteins over many eras, not years, over many years. Era means, of course, in monkeys also, monkeys are our ancestors. Monkey also histone protein sequence same. The histone proteins play a crucial role in maintaining structural and functional integrity of chromatin. So structural and functional integrity of chromatins done by histone proteins. The histone proteins are also important in the nucleosome model and nucleosome model of DNA organization. I will tell you that what is the nucleosome model and what is the solenoid model. So this is, so anyway, 5 types of, I will tell you how they are arranged. First of all, they are 5 of H2, H1, H2A, H2B, H3, H4. They are much conserved, no alteration from generation to generation because there is no change. They are firmly attached with DNA, acidic DNA. The E-stone is alkaline nature. Generally, E-stone alkaline and DNA acidic. The both are firmly attached to each other, right? And next one, non-histone protein. The non-histone proteins, they completely opposite, completely differ histone proteins. The simple one word, the non-histone proteins, what are the not, which proteins are not involved in heredity? These are all non-histone protein. What are not involved in heredity? They are all called non-histone proteins. The best example non-histone proteins, some of the enzymes, and during cell divisions, spindle fiber formation also protein. They also non-histone because the spindle fiber not hereditary, the enzymes also not hereditary, so they are non-histone protein. And another most important thing is the non-histone proteins display more structural diversity compared to histone proteins. This diversity is not conserved, not one generation to another. It is not only one generation to another generation. Within the cell, within the one organism, different tissues, also different makeup. For example, leaf tissue, one type of non-histone protein. Stem tissue, one type of non-histone protein. And fruit, another type of non-histone protein. The histone highly conserved. This is ancestor from the ancestor. It is stable without much changeable. Whereas non-histone proteins are they are clearly altered, not only one generation to another generation. Within the organism, one cell to another cell, one tissue another tissue is also different. Right? Non-histone proteins present in chromosomes are found to be ones which play an important role in condensation and movement of chromosomes. Already told you. Chromosome condensation I will tell you later. And movement of chromosomes during cell division. The rest of the non-histone proteins are enzymes which are involved in DNA replication, transcription. I will tell you later DNA replication and transcription. Right? This is histone, non-histone proteins. Now let's see how the arrangement. Just wait. Is it visible to you? Hello?
Yes, sir.
Right. Here given example human being, of course, human beings body, body, animal, tissue, cell, nucleus, inside the nucleus chromosomes. What are present inside the chromosome? Thread, elongated thread. This is called chromatin. Already told you what are present inside the chromatin: RNA, DNA, histone proteins, and clear DNA present. Now this is elongated DNA compactly arranged with help of protein inside the nucleus, inside the chromatin, inside the chromosome, in nucleus and cell. Right? Now see the arrangement. Is it visible?
Yes,
Hello?
sir.
Yes, sir. Yes, sir.
Right.
Yes, sir.
Right. So simple already told you in arrangement proteins, histone proteins, how they are, and non-histone protein, how they are, and what are the function of histone and non-histone. Now let's see how they are packing. Packing of DNA in the chromosomes. So this is altogether form a chromatin and altogether form a chromosome, right? So previously it was suggested that 4 parallel DNA histone fibrils are associated to form the chromatid of a chromosome. That is initial view. But later in Vicia faba plants, the packing of chromosomes, packing of DNA identified. But in this, based on this, there are several models are come forwarded to explain the arrangement of genes, arrangement of DNA. The most acceptable model is nucleosome model. Most one nucleosome model. First nucleosome model. Nucleosome model. And another one is solenoid model. The first nucleosome model, the discovered by, was proposed by Korenberg, and named by Howdett. So this nucleosome
Hello.
model, why? Tell me. Hello? Hello? Any doubt?
No, sir. No, sir. No, sir.
Right. So for revealing the arrangement, organization of DNA, first one is nucleosome model. Right? In eukaryotic nuclei, the long DNA molecules are compactly packed with the protein containing beaded-like structure. This beaded-like structure called nucleosome. This is one bead. This is second bead. Like this, it is the entire DNA. Throughout the DNA, number of beads are present. Due to the beads, they are compactly arranged. So this packed with help of protein. Already told you, packed with help of protein. Such proteins are called histone proteins. What are the histone proteins? How many types? Already told you. Can you repeat it?
5 types.
Anyone else?
Oh, 5 types. 8 and 5 types. Sir. H1, H2,
5
H2,
types.
A, B.
Right. Right. H1,
H2B,
H2A, H2B, H3, H4. Right. We can see here H3, 1, H4, H3, H2A, H2B. Right. So these histone proteins play a very important role in packing along DNA molecules in an orderly manner in a relatively small nucleus. In a small nucleus, the nucleosomes appear like a beads and a long chain due to the presence of histone protein. Let's see the structure of nucleosome. Each nucleosome is a disc-shaped structure consisting histone protein. Here, remember, the disc consists of 2 copies, 2 sets. That means here already told you H2, H1, H2A, H2B, H3, H4, 5. But here H1 the single and rest of the all, that means H2A, H2B, H3, H4. Therefore, and forms the octa. What is octa? What is octa?
8.
8. So these 4 histones H2A, H2B, H3, H4 doubled. Thus a total of 8 histones containing octamer from the core of the disc. Generally, double stranded DNA makes 2 complete turns around the histone octamer disc. Actually, not exactly 2 complete, 1 3/4 turn. That means actually how they are packed. For example, this is H2A, H2B, H3, H4. How many are there?
4.
How many there? H21, H2A, H2B, H3, H4?
4 only.
Right,
Five.
4 only. Just let me complete first of all. Just only 4 histone proteins. And this is 4 histone protein. Backside also same arranged. Backside also. Same H3, H4, H2A, H2B. Now how many?
8.
8. So this is called, this 8 one called as a octamer. Octamer disc. Octamer disc. Then this octamer is wrapped by DNA. Let me take another color. Right, blue color. One turn. Second turn. But second turn not complete. 2 turns. Incomplete 2 turns. That means one complete ring, one complete turn, and second 3/4, not completely. Right? So blue color is DNA. Remember, don't get confused. Blue color DNA and red color histone protein. So one is one histone protein loss. What is the missing histone protein here?
H5.
H1, right? Now this H1 histone protein. Let me take another color. H1 histone protein sealing the DNA strand. First of all, entered one turn. After second turn, now it is sealed with help of H1 protein. Sealed with help of H1 protein. And remember another thing, when compared with others, that means the rest of the histone protein, this H1 protein is not a constant, as such as H2A, H2B, H3, H4. Clear? Now this DNA strand again goes and take another disc. This another histone protein, octa-core. This blue color DNA, and again turns 2 rings, and again leave. Where the leave? The leaving place again closed with help of, sealed with help of
H1.
right, H1 protein, like this. So this is the package. You can see in diagram also here, this light violet color round balls, these are all histone proteins. They are histone protein. They are histone protein. How many? H3? 2 H3s. Remember, 1, 2. 2 H4. H2B 2. H2A also 2. Right? Actually, here this closing region. Here supposed to be H1. Here also supposed to be, sorry, here. Right, okay. H1. Right? Now this is called as nucleosome. This is called as nucleosome. This one. What are present inside the nucleosome? Hello?
Histone proteins.
What are present inside the nucleus? Only histone protein. Only histone protein.
Histone proteins and
DNA along with DNA, which is
DNA and histone protein. Both
Yes.
DNA and histone protein. Then these 2 terms, of course, remember, not a complete 2 turns, one 3/4. So for our convenience, it is 2 turns. These 2 turns are tightly held by H1 histone protein, tightly packed with help of H1 histone protein. The 2 turns of DNA, which turns the disc, generally contain 146 nitrogen bases. What are present inside the DNA? In further, if we reveal the DNA, it is completely filled up nitrogen bases, right?
Yes. Nucleotides.
Adenine, guanine, thymine, cytosine, they are the nitrogen bases.
Adenine, guanine.
In a 2 turns, the total nitrogen bases are 146. In this, in 2 turns, 1, 2, 1, 2. These 2 turns, the total. This is complete turn. This is incomplete turn. The total nitrogen bases within the DNA stand 146 nitrogen bases. The DNA extended. That means it is continuation. As continuous thread from one nucleosome to another nucleosome. This is one nucleosome. This is second nucleosome, right? First, second nucleosome. So these 2 nucleosomes connected with the DNA. This is called as linker DNA. This is linker DNA already clearly given. This one. This is linker DNA, right? Here the linker DNA 54 nitrogen bases consist 54 nitrogen bases. Almost in a nucleosome, how many nitrogen bases are there? In one nucleosome. To write exactly 200 nitrogen bases are there. So this is nucleosome model. Is it clear? It's very important in genome organization. Simple genome organization, how they are organized inside the chromosome chromatin material. Before that, you should know about the chromatin chemical structure. And next, how they compactly arranged inside the chromatin or chromosome. First one, nucleosome model. Is it clear? Hello?
Yes, sir.
Yes, sir.
Right. The second one, solenoid model. Solenoid model. The simple, remember one word. The solenoid model, the very compactly arranged. That means we can't observe the linker DNA space. We can't observe linker DNA space. These 2 histone, 1 histone, second histone, closely arranged, come close each other and compactly attached. Compactly attached and appear as a like this. For example, this is 1 histone protein, second, third. DNA stand, linker DNA, 2 turns DNA, linker DNA. So this is 1 nucleosome, second nucleosome, third nucleosome, right? Solenoid model, simple, they are arranged. Like this. What you observed in this?
Beads are arranged chain-like structure.
Right.
Linker DNA absent, sir.
Generally, the flowers weaved
Not
in
absent
these
here. The
two
linker DNA also present, but is very compactly,
Okay.
very reduced. Okay. That means then what you observed?
Beads.
Anything
The
else?
DNA cannot be distinguished. It's not clearly get separated or identified.
Any difference in
Compactly
lengthwise?
arranged. Closely
Right,
arranged.
compactly arranged, closely arranged. For example, for this is 5 meters length. This is 20 centimeters. That means very compactly arranged. So that arranged inside that tiny cell, very minute cell. Of course, Do you know one thing? That DNA length almost some kilometers.
Yes.
Then what about the cell? Are you able to see cell?
No.
No.
Some of the cells we can't. Some we can.
Give me example.
Egg cell we see, sir.
Egg cell.
Hmm.
Have you seen ever which egg cell?
Yeast we can, not yeast, not yeast. Some,
Give me any one example. What type of cell you directly identified with your naked eye without using any microscope? Almost all cells are microscopic. We cannot observe under our direct eye. Then what about the DNA? Actually, if you spread the DNA almost some kilometers, that means more than one human being height. Human being just a feet. But the DNA, if we spread the DNA, it almost some kilometers.
2.5 kilometers.
Yes. Do you have 1 kilometer even? A human being.
Sir.
Of course, the total an organism, not even a kilometer, not even a meter, that too in a cell, that is a very minute. Just imagine the very elongated DNA, the closely compacted, arranged inside the cell. How it is arranged? How the elongated thread compactly arranged inside the cell, inside the nucleus. The simply with the help of proteins, histone proteins. These histone proteins are elongated DNA, makes it very short. Make it very short with the help of histone proteins. So that compactly arranged inside the cell. Right? So solenoid model, in solenoid, this is difference, nucleosome solenoid. Hope you understand difference between this both. Are you understand?
Yes, sir.
Right. In metaphase and interphase nuclei, the much coiled chromatin is seen. Much coiled. The form of fiber, which is almost 30 micrometers, I will tell you 10 micrometers, 30 micrometers difference. Closely packed in nucleosome. Of course, nucleosome, the simple definition, solenoid model, the closely packed. Okay?
Okay, sir.
In this 6 nucleosome per one turn, 6 nucleosome, that actually here the 3 individual, 1, 2, 3. And again another 3 also, total 6. Total 6 nucleosome per one turn. 1, 2, 3, and again 3. This way. 1, 2, 3. Total 6. This 6 appeared as a one turn. In nucleosome, this is one histone protein, but in solenoid, like this one, 6 nucleosomes considered as a one. In nucleosome, only one. This is one nucleosome, one unit. That means one unit. If we take solenoid model like this, the 6, 1, 2, 3, 4, 5, 6 together only one unit. That means it would be one turn. Such many closely solenoid structure packs the entire DNA, packs the entire DNA of the chromosome. In this type of packing, at no point the DNA is buried. Instead, it is freely exposed all along the entire surface of the disk for genetic expression. Thus, nucleosome seems to be the universal device to compact the long DNA. Right? So that means the what nucleosome somewhat lengthwise, but solenoid this very closely compactly arranged. Very closely compactly arranged. Right? For example, now this is the genome organization. Just to 1, 2, 3, 4, 5, 6 points enough. 6 steps. 6 steps. Right? So genome organization, already told you. Now tell me, where is the genome? How it is organized? You said answer already. Where is the genomes? Hello.
DNA, sir. The nucleus
Where is
DNA.
Where is DNA?
Chromosome.
Chromatin, chromosome, chromatin,
Right,
chromatin.
DNA chromosome. Right. Okay, okay, okay. Chromosome. This is so we said this is chromosome. The chromosome made up of already told chromatin.
Yes.
Chromatin. This chromatin thread shows the folding with help of histone proteins. And this is solenoid model, and this is nucleosome model. This is DNA. First of all, genome organization means before genome organization, we already learned genome definition. Definition. A gene active site of DNA. That means you have taken DNA stand. This is DNA double helix stand. DNA double helix stand. The active, non-active portion also present. Okay. This is only pure DNA. DNA with histone protein. This is DNA with histone proteins. Already told you, already clearly explained about this model also, nucleosome model. In only DNA, the thickness is 2 nanometer with histone proteins 11 nanometer thickness. And just imagine, of course, it is equal length of the length. This is DNA equal length, nucleosome equal length. These are all equal length, but actually DNA elongated strand. When it is wrapped with help of histone protein, somewhat decrease in size. For example, this DNA first one. This is nucleosome model. Okay. And next one, solenoid model. And this beading shows the fold, shows the folding. This is one solenoid. Here 1 6, another 6, 1, 2, 3, 4, 5, 6. This is another solenoid. Next, this solenoid shows the foldings. That means it is highly reduced like this. So the increasing of foldings as a result decrease. That means lengthwise decrease, thick increase. Here 2 nanometer thick, 11 nanometer thick, 300 nanometer. Actually, this is not a 300 nanometer, 30 nanometer. Sorry, this is mistakenly 30 nanometer. And next one 300 nanometer, 300 nanometer. That means thick one increased. Next 700 nanometer. Next chromosome. And finally, it is appear as this beadlets appear as a chromosomes 400 nanometer. So first start DNA based on strings, that means beads, chromatin packed. These also beads appear beads, but closely packed beads. the beads level increased, condensed form of chromosome, and finally chromosome clear true chromosome form. This is genome organization. Is it clear? Hello?
Yes, sir.
Is it clear or any doubt?
No, sir.
Yes, sir.
Right. Right. Now let's see the chromosome morphological structure. Now the chromosome. Can you define the chromosome? Can you
Chromosomes
define
are the
chromosome?
vector of heredity.
What?
Vectors of heredity.
Okay, heredity.
Vectors. Vectors,
Okay,
sir.
this. Okay, vector. Next, how it is look like?
Means
Rod shape. So chromosomes are found rod-like structure present in nuclei of cell. The who observed the first time this rod-like structure? This rod-like structure were first observed by Carl Nageli.
yes, yes.
Carl Nageli. Can you rough it?
Okay, sir.
Good is. This way. కూర్చో. రానా chromosome structure morphologically it can be observed very clearly during the cell division, particularly in metaphase. Just wait. Where it is? Right. Downloaded where it is. Right. Chromosome structure Already told you chromosomes rod shaped hereditary vectors. First observed by Carnegie. The morphological structure and size of chromosomes can be observed very clearly during the cell division, particularly in metaphase and
Yes.
anaphase stages also. Metaphase and anaphase. Actually, different. Don't talk, please. Hello, please be silent. Right, mainly the morphological structure can be observed in metaphase. The shape of chromosome can be observed in anaphase.
Anaphase.
Right. For studying the chromosome morphology in the mitotic divisions, generally, no need to this all. Okay.
I
Right.
think Satan and Baveri have studied this.
Right. So mainly the clear components of the chromosomes: chromonimata, chromomeres, chromatid, centromere, and it's not given here, telomere, secondary constriction, so on. And this chromosome covered with small pellicle layer.
Yes.
Right. And short chromosomes also part of the chromosome in some organisms.
Yes.
Right. What is the different chromonimata, chromomeres? Already told you, first is the part of chromosome chromonimata and chromomeres and chromatids. They are interchangeable. First, let's see chromonimata. It is a much coiled chromatin.
Yes.
First of all, for example, this is chromosome with two chromatids, right? The based on chromatin structure, it is given the different name: chromomeres, chromonema, chromatids. Chromonimata, coiled chromatin thread, present in the interface of the cell. This loosely arranged chromonimata gets condensed. Or simple chromonimata.
Sir,
The voice is not clear.
cut out on the voice.
Okay, just wait. All right. Are you able to see screen?
Mm-hmm.
No, sir.
Yeah,
Yes.
now
Screen
Yes, sir.
screen is clear or not?
Clear, sir.
Okay. Right. Simple here. First chromonimata. Chromonimata. chromomeres bead-like appearance like this. This is just the structure of chromatin threads. Chromatin threads loosely coiled like this, loosely coiled. This is chromonemata, or somewhat straight, like somewhat straight. It is clearly straight. It is chromatin, but it shows that some foldings. That is the called as chromonema, and chromoneres completely beads like a chain, like this, like a chain. But this is due to the condensation. Condensation means increasing the folding. Increasing folding. For example, for our better understanding, this is nucleosome model, this is solenoid model, and these foldings deeply increased and reached the thickness, very thickness. This is called chromatid. This is called chromatid, like this. Chromonema, chromatin, loosely beads, chromonema, closely beads, chromomeres, and these beads increased thickness. Now form the chromatid. Form the chromatid, right? So now it is. Hello? Is
Yes,
it
sir.
clear?
ఇంకొకసారి
Chromonema,
చెప్పండి.
chromomere, chromatid. Simple. Already you know the chromosome package. Solenoid model, nucleosome model. First of all, in nucleosome model, DNA with histone protein, loosely packed, right? When it is loosely packed, that is called as chromonema. Chromonema, like this.
Just like
Chromonema.
jasmine weaving, sir.
Just like
Jasmine weaving. See, sometimes we weave with the thread. Sometimes we weave with the help of a needle.
okay. The help of
మళ్ళీ పూలు అల్లడం వేరు ఉంటుంది, సూదితో కుచ్చితే కంపాక్ట్ గా దగ్గరికి వస్తుంది. The thread like structure we can compare with the chromonema. Sorry, this
now here the flower compare with
నో సార్, లైక్ జాస్మైన్ మల్లె పువ్వులు ఉంటాయి కదా సార్, అవి థ్రెడ్ తో అల్లినప్పుడు ఇలా చేత్తో అల్లుతారు. అదే సూదితో కుచ్చినప్పుడు దగ్గరికి వస్తాయి, కాంపాక్ట్ గా వస్తాయి.
అదేనండి మీరు అన్నది.
ఫ్లవర్ బీడ్స్ కంపేర్ చేస్తుంటారు.
ఫ్లవర్ బీడ్స్ మీరు ఇచ్చిన ఎగ్జాంపుల్ లోనే ఫ్లవర్ డిఎన్ఏనా హిస్టోన్ ప్రోటీన్ అడుగుతున్నా.
హిస్టోన్ సార్, హిస్టోన్ ప్రోటీన్.
రైట్.
డిఎన్ఏ ఇస్ a thread like structure.
రైట్. అట్లా మీరు కొంచెం ఇమాజిన్ చేసుకోండి. దాంట్లో అంటే థ్రెడ్ లాగా కొంచెం కనిపించి బీడ్స్ తక్కువ ఉంటేనేమో క్రోమోనిమా. బీడ్స్ ఇంకంటే కొంచెం కంపాక్ట్లీ అరేంజ్డ్ అయితే బీడ్స్ ఇంక్రీజ్ అయితే క్రోమోమియర్స్, బీడ్స్ అపియరెన్స్. Chromomeres are formed due to the tight folding of DNA. Remember it. Tight folding of DNA in chromosomes. It clearly observed in interface of chromosome. Okay. In metaphase, the chromomeres are not visible because the chromosomes themselves are condensed state. And finally, it is reached the chromatid. Hello?
Yes, sir. Yes.
Okay, sir.
Is it clear or not?
For me, it's clear.
Don't get confused with the one word. That don't get confused with one word is chromatid. Of course, you should know the chromosome main component, pellicle layer in the chromatin. That's it. Chromatin thread. Just wait. For example, this is chromosome chromatin threads. No, chromomeres. Chromomeres, chromosomes. Elongated sets, some loosely arranged beads. Chromonema. This is called chromonema. Next. Chromomeres. Chromomeres. These chromomeres are formed due to tight folding. Right? This is called chromomere. They are highly
I had drawn that. So
tell me.
I'm showing the chromatin structure.
Is it clear?
Yes.
Now it is called chromatid. Okay.
Okay.
Here these chromonema, chromomere mainly appear in interphase and also prophase also somewhat. But this chromatid clearly appear in metaphase. Metaphase, it is clearly appear in metaphase. So this is only chromatid. Is it clear first? Hello?
Yes, sir.
Right. Just imagine this elongated, beaded, much beaded. That's it. Okay. Right. So another part is centromere. Where the centromere?
In the
Where the
center of the
centromere?
chromosome.
Center.
Center. This is centromere. This one, the right one. This is centromere. Centromere. Primary constriction. Centromere or primary constriction. Right? It is a darkly staining heterochromatic part. I will tell you what is heterochromatin and homochromatin, which is present between the arms. This is one arm. This is entirely one arm. This is another arm. So it is located between the two arms of chromosome is called centromere. It is also already told you called as primary construction. On the either side of the centromere, highly repeated DNA. Either side of centromere, this side and this side, highly repeated DNA is present, which stains darkly. I will tell you when I explain about the heterochromatin, homochromatin. The center of centromere specific DNA sequence and proteins together form a disc. That means this centromere within the DNA specific sequence, that is the repeated DNA sequence, and covered by protein structure. This is called kinetochore disc.
Break on sir voice.
Not audible, sir.
Now clear?
Yes, sir.
Now clear? Right.
Yes.
So the centromere which consists of specialized DNA order and it covered with special protein that is called kinetochore disc, right? The kinetochore appear as a trilaminar, 1 lamella, 2 lamella, 3 lamella, laminar structure. The outer region proteinaceous convex disc. The function of this convex disc just attachable to the microtubules or spindle fiber. The kinetochore disc function, only implementation or attachment of the spindle fiber during cell division in anaphase. So in the cell division of anaphase, the spindle fibers are formed. They are firmly attached at kinetochore disc, right? So this is the primary constriction and kinetochore disc, right? And next one, secondary constriction. Along with the primary constriction, somewhere not exactly middle, somewhere the secondary constriction are present. From the secondary constriction, nucleolus generated, nucleolus formed, and just wait. The secondary constriction.
The voice breaks.
Now the device visible. Hello.
Yes, sir.
Right. So this is secondary constriction. Secondary constriction, centromere, primary constriction. The somewhere secondary constriction present from this region, nucleolus developed, and end of the chromosome, that is the sat DNA and called satellite, and this blunted end called as telomere. The telomere function one chromosome not to attach with another chromosome. That is the function of telomere. The satellite chromosome, the satellite portion function unknown so far. So this is morphological structure of chromosome. So chromosome morphological structure different, one question, and genome organization also question. In genome organization, you can explain about the solenoid model and nucleosome model, and clear diagrams also. Okay.
Okay.
Is it clear? Any doubt in this?
No, sir.
Okay.
If you have any doubt, ask me.
So polarity is shown by telomere.
Yes, polarity. This blunted end, the blunted end. So this is called telomere. Okay. And this is pellicle, not given here. This layer is called pellicle layer.
The outer layer.
Right?
Yes, sir.
Right. So far we completed genome organization and chromosome structure.
Yes, sir.
Okay. Thank you, one and all.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.